¿Sabías que tu cuerpo tiene un termostato interno que decide cuánta energía quemas y cómo te sientes cada día? Ese termostato es el sistema endocrino y su gran director de orquesta es la glándula tiroides. Esta pequeña glándula produce hormonas que viajan por tu sangre para indicarle a tus células a qué ritmo exacto deben trabajar. Pero, ¿cómo se conectan el cerebro, la tiroides y tus genes a nivel microscópico? Acompáñanos a descubrir el viaje exacto de estas hormonas para encender tu metabolismo.
To maintain a stable internal temperature and a consistent metabolic baseline, the human body requires a strictly regulated communication network connecting the brain to the endocrine system. This diagram illustrates the macroscopic infrastructure of that network, the hypothalamic, pituitary, thyroid axis. The cascade begins when the hypothalamus releases TRH, stimulating the pituitary. The pituitary responds by secreting TSH, the primary signal establishing the metabolic set point, driving the thyroid to synthesize hormones. Because TSH has a relatively long plasma half-life of about 50 minutes, its levels remain highly stable in the blood. This stability makes a single TSH measurement a highly accurate clinical marker for diagnosing thyroid dysfunction. Systemic metabolism, therefore, is not a decentralized process where individual organs guess how fast to run. It is strictly dictated from the top down by the pituitary's TSH set point. At the pathway's base, the thyroid gland produces two effector molecules, T4 and T3. Once in the bloodstream, they travel to peripheral organs and circle back to the brain. Here, high concentrations of thyroid hormones rapidly suppress TSH expression, cutting off their own production signal. This inhibitory feedback is mediated by a specific nuclear receptor isoform known as TR-beta2, which is selectively expressed in the hypothalamus and pituitary. The entire loop functions much like a mechanical thermostat. When the TR-beta2 receptors in the brain detect sufficient metabolic heat from circulating T3 and T4, they immediately throttle the TRH and TSH signals. To understand how this large-scale system controls local tissue down to the cellular level, we have to look inside the specific follicular factory that builds these effector molecules. This reveals the polarized thyroid follicular cell. TSH docks at the basolateral receptor, initiating a CAMP cascade. The NIS channel actively pulls iodine into the cytoplasm. Simultaneously, the cell synthesizes thyroglobulin. At the apical border, thyroid peroxidase couples iodine onto thyroglobulin, creating T4 and T3. Finally, the polarized cell reabsorbs this coupled protein complex and breaks it down through proteolysis. It then releases the mature T4 and T3 molecules out the other side and into the bloodstream. Systemic metabolic control relies entirely on this mechanical assembly line, meaning the entire endocrine cascade is strictly limited by the physical availability of dietary iodine. Once synthesized, T4 and T3 travel through the systemic circulation to reach peripheral target tissues like the heart, liver, and muscles. To exert any metabolic effect, these circulating hormones must physically enter the target cells. However, they do not simply slip through the cell membrane via passive diffusion. Cellular entry requires specific active monocarboxylate transporters, such as MCT8, to pull the hormones from the extracellular space into the cytoplasm. The clinical necessity of this active transport is absolute. Patients with mutations in the MCT8 gene cannot properly absorb thyroid hormones into their neural cells, resulting in severe psychomotor retardation. Once safely inside the cytoplasm, the primary circulating hormone, T4, is actually biologically inactive. It must be chemically modified before the cell can use it. Peripheral tissues express specific enzymes that deiodinate T4, stripping away a single iodine atom to biotransform it into the highly potent T3 molecule. Because of this required conversion step, peripheral organs are not passive targets. They exert direct control over their own metabolic rate by regulating exactly how much T4 they convert into active T3. This diagram tracks the newly activated T3 molecule moving from the cytoplasm directly into the nucleus of the target cell. Inside, it seeks its ultimate target, thyroid hormone receptors, already bound to the promoter regions of metabolic genes. These receptors do not operate in isolation. They form heterodimers with the retinoid X receptor, allowing them to lock tightly onto specific thyroid response elements within the genetic code. To understand how T3 activates these genes, we first have to look at the receptor's unliganded state. In the absence of a hormone, the receptor complex actively binds to co-repressor proteins. These co-repressors physically disrupt gene expression. They sit on the DNA and act as a biological brake, shutting down the cell's metabolic machinery. The baseline state of the thyroid receptor is therefore not idle. Without T3, the receptor actively and structurally represses cellular metabolism. When T3 finally arrives, it physically docks into the binding pocket of the thyroid hormone receptor. This physical connection induces a massive conformational change in the receptor's overall structure. As the receptor changes shape, the co-repressor complex can no longer hold on. It is forcefully disassociated and expelled from the binding site. With the co-repressor out of the way, the new shape of the receptor immediately attracts and recruits coactivator proteins. The docking of these coactivators completely releases the cellular brakes, driving heavy transcription of the target metabolic genes. T3 operates as a bimodal switch. It does not simply press a metabolic gas pedal, it must first physically detach a biological brake to initiate transcription. These strict nuclear mechanics directly explain the clinical presentation of a patient suffering from hypothyroidism. When researchers delete the genes coding for these thyroid receptors entirely, the physical effects are actually less severe than a simple hormone deficiency. That is because in a hormone-deficient patient, the receptors are still on the DNA. Without T3, their target genes do not quietly idle, they are actively locked down and silenced by co-repressors, resulting in a profound drop in metabolic function. Exogenously replacing thyroid hormone sends T3 back into the nucleus. It displaces the co-repressors, lifts the genetic blockade, and restores the HPT set point. Regulating human metabolism requires a seamless, continuous cascade. It begins with a macroscopic set point in the hypothalamus and ends with the precise physical displacement of a single repressor protein on the DNA.